Thank you, Ali. Good morning, everyone, and welcome to the PMD session. I will introduce the session as you saw it yesterday. We have a colleague from Germany, Gesine Saher, who will tell us about disease models and pathophysiology as one of the key successes. One of the challenges is how do we plan, okay, is it better now? Yes. Good. So Chloe Stutterd from Australia will talk about the challenge of planning clinical trials in an ultra-rare disease, because some things are up and coming. And we heard yesterday already several times the challenges we see, for example, relating to geographical locations. Jigyasha Sinha from Kolkata will talk about the challenges in the developing world. I thank the workgroup members who all made time to contribute to that session. So I think Pelizaeus-Merzbacher disease, or PMD, is perhaps even the oldest leukodystrophy. It was described in 1885 in a beautiful long clinical description by Pelizaeus, and in 1915 the neuropathology was elucidated, and also it's a beautiful paper with a lot of illustrations, and the myelin lack and the fact that it's an X-linked disease were already recognized then. We have quite a range of phenotypes, from conatal Pelizaeus-Merzbacher disease presenting right after birth to milder form spastic paraplegia type 2. It is said that the most frequent genetic variation, is duplication of the PLP1 gene. Otherwise, we see missense mutations, deep intronic variants, and deletions. I think the latter ones are quite rare. Typically, clinical presentation is in early childhood or infancy with nystagmus, developmental delay, hypotonia progressing to spasticity, and ataxia. Heterozygous females may manifest mild to moderate signs of the disease, usually from adulthood. There is some genotype-phenotype correlation, but it's still difficult if we have a PLP1 variant to predict the disease, if it's a private variant. And perhaps I should mention that all, more or less, all genetic variants in PLP1 are pathogenic, because it's an extremely well-conserved protein. There is an active PMD community, a large patient population where it's not one of the ultra-rare diseases. We have rarer leukodystrophies, of course, but it's a rare disease. We have a clinical heterogeneity, but we still lack biomarkers, and MRI biomarkers perhaps are a good way to monitor treatment successes. There is one planned clinical trial repurposing diferipron in the Netherlands, in our center, and there are approaches to start a treatment trial with antisense oligonucleotides. I think that one of the biggest challenges is that we have those symptomatic female carriers who are often not recognized as having symptoms due to their PLP1 variant. I think that's very similar to the situation in adrenoleukodystrophy, and also the clinical presentation is very similar. Newborn screening is not yet thought of, and we still need disease-specific standards of care in Pelizaeus-Merzbacher disease. The Heatmap regarding the disease-modifying therapeutic pathway is relatively green, so I was glad to see that, because in the next session about 4H, that looks really different. But we have not yet access to disease-modifying therapies. And I think the access to diagnosis is very variable depending on in what part of the world you are in. So I would stop here to keep it short and ask Dr. Saher from Göttingen to tell us about the disease models.
So, good morning again. Thank you for inviting me to this very stimulating, can you hear me all right? Yeah? No? Session or meeting. Thank you for organizing this. Thanks to Omar, thanks to all of this organizing team. And thank you, Nicole, for giving me the opportunity. So I'm a researcher from Germany at the Max-Planck Institute in the laboratory of Klaus Nawe. And I know that you don't need any introduction to myelin. It's just two things I want to emphasize here. So if we talk about myelin, we also talk about lipids because, and especially cholesterol, because 70% of the brain cholesterol resides in myelin membranes. And moreover, particularly it's PLP that directly associates with cholesterol. So you cannot, if you have a problem with PLP, you often have a problem with cholesterol. And the other thing I wanted to emphasize here is that, of course, you all know that myelin is there to allow saltatory impulse conduction, but it also reduces the energy demand of axons to fulfill this task. And this is often a problem, I think, when it comes to neurodegeneration or damage to axons, that there could be an energy problem. Okay, you know, Nicole has told you this, and also that the mutations in the PLP gene can be very variable. I mean, there's a large spectrum of disease or of mutations and also of disease presentations. And this, to model this in animal models, this led to the development of several models for conatal and more milder forms of the disease, and several mutations have been introduced, many of which were spontaneous mutations, so they were just being found. We have models in different species like mice, rats, rabbits, dogs, monkeys, and of course also patient-derived material will be instrumental in elucidating the mutation-specific pathology. So we have, although we have so many models, each of these models only different aspects of the human disease, and none of these models will model the entire disease, which is, I mean, likely and has been alluded to yesterday as well for other diseases. But nevertheless, they are instrumental in understanding the mechanisms of pathogenesis in these models, and then, of course, for preclinical trials, evaluate the safety and efficacy of potential drugs. So when we have a closer look at the myelination process in the normal wild-type situation in mice and also in men, PLP here in this blue dot associates with cholesterol, and they get transported to the plasma membrane and then to the developing myelin sheath. And in this cross-section to the spinal cord of a normal mouse, you see there's myelination. And this is very different in case of a PMD mouse model, in this case, an PLP overexpression mouse, which models the duplication situation most closely. So you see here that there's no myelin, and most of the PLP is residing within the cell. So in case of the overexpression situation, this leads to an accumulation of PLP in the cell linked to an accumulation of cholesterol within the cell. And this perturbs the homeostasis also of lipids and of cholesterol trafficking within the cell. In case of other mutations of the PLP, the relevance of the cholesterol homeostasis might not be so pressing. However, what you can see is that not as wild-type PLP that gets transported to the plasma membrane here in cell culture, many of these mutant PLP forms remain retained intracellularly, and in this case here in the ER. And this, of course, induces a stress response in the oligodendrocyte. So there are, of course, mutation-specific stress responses, and we are far from understanding all these differences here. But what you can see is that there is cell stress, and sooner or later, it's very likely that this will lead to a loss of oligodendrocytes. And coupled to this, there is poor trafficking of PLP to the myelin sheath, and we have poor myelination and thin myelin sheaths. And this is coupled, of course, to the stress response here. So, but this is not the end of the story. Although we quite maybe understand the main processes in the PMD oligodendrocytes, this of course will lead to an inflammatory gliosis triggered by the loss of oligodendrocytes, the loss of myelin, because the PMD myelin is often not stable. So we have here a stress response, and this can, of course, add to the problem, because with disease chronicity, also microglia the microglia cleanup of debris will, exhaust. The whole system of this repair will exhaust. And then finally, this is observed in mouse models as well as in patients, that there's that this whole situation damages the axon and also the neuron. And this is here shown in this electron micrographs, where you can see here in this high pressure material these like innocent looking circles. These are the mitochondria, and what you can see here in a very young PMD PLP overexpressor mouse, that the mitochondria profiles are enlarged, they are swollen, and this is also, or this often reflects the functional damage of these organelles, and lead finally to accumulation of material in the axons and neurodegeneration. So if we look at this disease situation more closely, what could be potential strategies to ameliorate PMD? Coming from this cell biology point of view, we can, of course, go down right to the core of the disease, so correct the mutant PLP. And as Nicole already said, there are several strategies to suppress the gene expression, for example, which has been shown in mice by Ken, he's somewhere, I don't see him, and also by Paul's lab, Paul César's lab. There are cell-based strategies, so transplantation of stem cells or neurospheres. Then there's a large topic of providing support to the oligodendrocyte in addition to correcting the PLP. This is, of course, a really mixed bag. And depends a lot of the initial mutation in the PLP gene. So what one can do is improve the PLP trafficking to the plasma membrane, reduce the stress response, or at least help the oligodendrocytes to cope with this mutant PLP, and then prevent the death of oligodendrocytes. And there, as also Nicole said already, iron chelators have been found to be very effective in preventing the ferroptosis process of oligodendrocytes. And there's another means to support the PMD oligodendrocyte, which is the cholesterol supplementation. This stems from our lab and is restricted, I think, to duplication patients. And this is applied to at least one patient in the, which is hosted by Steffi Kulachewski, Rea Kulachewski in Göttingen. And I think it is crucial that also the I mean, the non-informal information is spread that, I mean, how do these patients cope with these treatments and whether it's, I mean, successful and to what degree. I think you have to talk more to each other and provide more information to each other beyond what you write into publications. And then there's a large topic, again, to support axons, neurons, and microglia. And there we have also developed a treatment, which is a ketogenic diet, and a couple of patients across the world are receiving now ketogenic diet. This is in, so from north to south. I know of a patient in Hamburg with, and in Göttingen is another patient, and also in Italy, in beautiful Milano. There are several patients and probably more to come. And so this is what our, I mean, how our research has, I mean, of all of us, has triggered the one or the other attempt to translate our findings. There will be, of course, more to come, and I would like to share with you just briefly an application of a cholesterol synthesis intermediate, squalene, here, which we have found in MS models, but MS models are in a way similar to leukodystrophies because they involve the loss and repair of myelin sheaths. And I don't want to go into detail, but the cholesterol synthesis pathway in microglial cells is really crucial to drive the repair of demyelinated lesions, and this is not mediated really by cholesterol, but by the immediate precursor, desmosterol, which then leads to an LXR response that does two things. It helps the cholesterol efflux from the microglia, which with disease chronicity often internalize so much cholesterol and lipids that they suffocate from all of this phagosized material, and it has also the potential to resolve the inflammation, which then can promote a remyelination. So we have found this in models of MS, but have also applied this to our PLP transgenic mice, and what you can see that it helps a lot in reducing the motor phenotype of our mice, and it also reduces the T-cell infiltration and the myeloid cells in the brain as well as in the spinal cord. This is remarkable because we had in another treatment arm, we combined the squalene together with the ketogenic diet, and this was as effective in these measures. And so what I can say to this, we are not at the end. There will be more ideas, more targets. There will maybe the combination therapy as something to think about, personalized treatments to the different mutations of PLP, new targets, and yeah, future will tell us. Okay, and then I thank you for your attention and hand over to Chloe.
Good morning, I'm Chloe Stutterd, a clinical geneticist in Melbourne, Australia. I'm grateful for the opportunity to present this morning on behalf of the PMD working group, and I'm going to discuss one of the key challenges identified in managing how to coordinate multiple clinical trials. So as we've just heard so beautifully, thank you, there are several potential disease-modifying interventions being identified in preclinical studies. And the fact that we've identified the coordination of trials as a as a key challenge reflects great progress in this field, which is exciting. So as we've heard, currently available and being trialled are the ketogenic diet, and this trial is based on the evidence that's been published in a mouse model where there was amelioration of axonal defects and promotion of myelination and improved motor function with the use of the ketogenic diet being a form of lipid therapy that ketone bodies being able to cross the intact blood-brain barrier, which we'd expect in this disease. And as we've heard, that's being trialled in a couple of countries, a couple of patients, as I understand, with a shared protocol, common endpoints, and a plan for, I think, primary endpoints being motor outcomes, and a plan for interim analysis after 2 years. And the other trial, soon to start in the Netherlands as a proof of concept trial in patients with the conatal form, trialling for 1 year the use of deferoprone, the small molecule iron chelator, and as we've heard, this is based on cell studies that identified hallmarks of ferroptosis in oligodendrocytes from affected patients, and then benefit of treatment of mice, of the mouse model, the Gympie mouse model, with deferoprone, and the observation that it rescued oligodendrocyte cell death, promoted myelination. This is a registered trial, and the primary endpoints being motor outcomes, secondary endpoints being myelination on MRI, and exploratory endpoints looking for biomarkers in the CSF and blood. And this recent publication, which showed benefit in the mouse model of suppression of PLP1 expression, and this was initially with the use of CRISPR-Cas9 gene suppression and then using an ASO, and in that mouse model there was observed improvement in myelination and extended life expectancy. So that's an exciting new avenue to pursue. So then this great progress poses the challenge of how to coordinate several trials simultaneously, and we heard yesterday we're not alone in this situation, but we need to think about how to prioritise which therapies to pursue in a trial, particularly if they're available therapies such as the ketogenic diet, then there's going to be limited funding to pursue these trials, and how to prioritise patients for each therapy and how to compare efficacy between these different approaches. So I've been involved in the PMD natural history study, the retrospective study run from CHOP, but I'm not currently running any clinical trials for treatments for PMD. So in considering this issue, I've drawn on experience from treatment of other monogenic diseases at our centre. Several of you will be familiar with the trials for SMA and know that there have been several trials running simultaneously for different therapies, and are now to the point where we're looking at trials for combination therapies, which is exciting. And in my situation as a geneticist in the molecular therapies group, to date a lot of our trials have been for skeletal dysplasias, because that's we have expertise in that field at our centre, and specifically for achondroplasia. I appreciate very different to PMD. For those that aren't aware, that's the most common genetic cause of short stature with life-threatening complications, but a much higher level of function, so, and very clear measurable endpoints, but I guess we can still learn from the experience of running multiple trials simultaneously for different agents. So currently there's 4 available therapies, and we're running at least 8 trials with different subgroups, different ages, different endpoints. So I think what I've drawn from that and from my discussions with our neuromuscular team, in fact, it was only that in fact, we haven't faced a lot of difficulty in identifying patients for specific trials when they become available because of the different timing, because of several issues. So these considerations are the specific mechanism of action of the intervention, its expected efficacy, meaning having endpoints, which endpoints are available to measure the mode of delivery and the burden of trial participation, and I'll speak a bit more to that. But in the case of SMA, in fact, it's only in the most recent application to ethics for a trial it was necessary to include the option that if there were several patients who were equally suitable to participate in the trial that exceeded the available number of places, then they would have the option to randomise that selection process. But that's the first time that that's actually needed to be included. So in terms of the consideration of treatment mechanism, we've heard that in PMD there are 3 distinct mutation types with a degree of genotype-phenotype correlation. And so obviously depending on the mechanism of the therapy, that may in itself limit the patients eligible to participate. In terms of expected efficacy based on preclinical trials, that may also inform which patients are going to be most suitable. Obviously, if it's a mutation-specific therapy compared with perhaps a approach to modify an endpoint such as mitochondrial dysfunction, then we would expect that the benefit is likely to differ between those, and we might prioritise different patient groups to those different studies. And most importantly, for all of these, we need meaningful endpoints that we can accurately measure. And in PMD, that's quite challenging, in particular for the most common form, the classic form, because of the slow disease process, but also because of the developmental phase that's seen, followed by the degenerative phase. So there's the risk in the developmental phase of false positives in terms of attributing benefit to the intervention as opposed to the development that's seen in that phase, the normal development, delayed, but there is development seen. And then in the degenerative phase, the consideration for, again, being able to appreciate benefit, but weighing that properly against the risk of the intervention and the burden of the trial at that stage of disease. The mode of delivery can also dictate which patients are eligible. A gastrostomy tube is, I would say, probably essential for the use of the ketogenic diet. It's otherwise a very challenging diet to adhere to. And for some patients in advanced stages of disease with severe scoliosis or previous surgery, there may be contraindications to lumbar puncture, for example, if that's required for intrathecal administration or for monitoring. Likewise, if a general anaesthetic is required regularly for MRI assessment and or intrathecal administration, then that may be a contraindication or an exclusion for patients with severe respiratory disease. Several other considerations that can also dictate patient participation can include the travel to the trial centre, certainly in our experience in Australia, which is the same size as continental US, obviously a lot less patients, but they're spread all around and we're the only trial centre. That's going to be a consideration, and I think we have a real responsibility to properly inform families of the burden of trial participation and ensure they do fully understand what's required to adhere to the protocol and to optimise retention in the study. And I think that's really our responsibility to ensure that we do select patients appropriately, considering the family context and how well they can manage participation in a trial as well as the care needs of their child, and that it's in the best interest of the child. And social context may be relevant. I'll be interested in people's thoughts. There may be cultures where patients put more pressure on clinicians for participation in trials when it may not be, in our opinion, in the patient's best interest. So that may be a factor also for us to consider. So in conclusion, I think we need to remember that the priority is for us to conduct high quality trials to accurately determine efficacy and safety of interventions, and first do no harm and make this our goal rather than treating our patients' desperation. I think in the long term we owe it. This is really our goal, and so we need to continue to focus on preclinical or proof of concept studies to select potential therapies with good evidence of or a good chance of efficacy, to focus on those. And for the situation where the therapies are already available and approved, I think it's critical that we collaborate on trialling those interventions with shared protocols, common consistent endpoints, and as the example was given yesterday for vanishing white matter disease, that's just absolute ideal model of the vanishing white matter disease consortium and the approach in MLD, where there is a collaboration in trialling these therapies, because we really need to avoid the risk of uncontrolled trials. We've seen that happen in other conditions where an available therapy is trialled in an uncontrolled fashion. Perhaps there's a false positive result. That therapy is then available to patients worldwide. There may be significant associated risk, and very hard then, when the community believes that that's beneficial, to ever go back and run a proper placebo-controlled trial and determine efficacy. So I think as tempting as it is to jump in and try these therapies, and we obviously all want to do everything we can for our patients, we really owe it to them to run these trials properly. So I don't think I've answered any questions, or solved any problems, but hopefully provided some food for thought and further discussion. Thank you for your attention.
Good morning and namaste to everyone, and I'm very glad today to be here because Nicole, being a very close friend of mine, she could actually help me out with this, because it's coming from a developing nation. It is very difficult and challenging for us actually, and since yesterday I have been seeing how therapeutics have really taken over, but for us even diagnostics is still a challenge. So with this at the outset, I would really thank the organizers, the GLIA committee, Mario and Nicole for giving me this opportunity to be talking here. I would also let you know that, just to get my visa, I had an appointment of February 2024. And thanks to Omar, if he wouldn't have been there, I would not have been able to make it to this meeting as well. So I'm really thankful to everyone. And with this, I would just start my talk of perspective of a developing nation on these rare disorders, especially PMD. So as we look at the developing nation, the demographics, the population demographics has always been that it's always, it's on like a sigmoid curve, it keeps rising and rising. However, the developed nations have it in a stable fashion, and that is where we, lack in terms of resources and everything. So when we talk about the Indian subcontinent, it caters to almost 2 billion population. So we can imagine how much of a disease burden we do have also in our countries. And when we talk about the Indian subcontinent, it consists of Bangladesh, Bhutan, Nepal, Sri Lanka, Pakistan, and India. And these people from these nations also go to each neighboring nations to get the treatment. Especially we have a huge load coming from Bangladesh and Bhutan to get treatment. So health has always been a very low priority for us. Hardly 1.2% of our GDP goes into the health sector, and we have been this is a very big challenge for us actually to fight it out for our patients. So when we talk about leukodystrophies in India, it is rather we have very limited studies on the spectrum of the leukodystrophies in India, and there have been 2 large studies published from 2 centers, and so in a 5-year study period done at All India Institute of Medical Sciences, we had about 80 confirmed cases of leukodystrophies, and we found that the major chunk comes from the MLC group. So because of the Agarwal community and the inter-community marriages, consanguinity and all those genetic factors, MLC remains very high on the list, and this is followed by metachromatic leukodystrophy and followed by PMD. So as we see, it's not that low in numbers, and this is also followed by adrenoleukodystrophy. And when we talk about PMD and PMLD in India, we have seen all sorts of mutations, whatever has been reported, but there are only isolated case reports in our country, and we have an unknown case burden still in our country. So how do we deal with our patients in our setup? So we have a major population from the village and a very small population, I would say about 60% comes from village and 40% from the city. And from the village, actually, 80% of them are lost in the village itself. So they are not able to reach the proper clinician also for a treatment. And that is mainly because of illiteracy, social taboo, being poor, etc. We are trying to bridge this gap by organizing camps, peripheral clinics. We are trying to reach out to them, but it is still a challenge for us. And they are usually seen by one general physician who caters to all sorts of clinical work. And from there, he might like to refer to a pediatrician who is often based in a city. And the pediatrician actually identifies the developmental delays and early signs, and from there they go to a neurologist or a child neurologist. Now here also we lack uniformity because the referrals may not be very integrated, they may be fallouts, and there is a large patient load. So in a day, a general pediatrician might be seeing about 60 patients also, and a specialist would be seeing around 30-40 patients in the outpatient department, so it is very challenging. And from the neurologist, actually, the way to approach the patient remains quite similar to the Western world. We do our investigations, but there remains the challenges of, again, the cost burden, lack of resources, genetic diseases are still not insured, and there is dearth of diagnostic experts, including that we do not have very good neuroradiologist access also in the major cities as well. From there, when we offer symptomatic care, it is with medications and rehabilitations, but then again for genetic counseling and genetic support, there is a lot of difficulties over there. We have very few experts over there as well. And then we have these regular follow-ups, which we do not have the support from the government, so we have a lot of defaulters there itself. We have very limited patient support groups in only certain diseases. We do not have it in leukodystrophy. That is on my mind still. And we do not have a registry. We have institutional registries still, but we do not have a registry in the country which is uniform. So some of the observations at my center where I work in last 2 years, this I have just put up because this is the amount of caseload that we have. I just picked up hypomyelinating leukodystrophy at my center, and we identified about 27 cases. This is an unpublished data. And we could identify 14 of PLP1, which is in which we saw there were 9 missense mutations. So initially, we were thinking that maybe duplications are more common, but I am also not very sure whether it's because that we go for the clinical exome sequencing that is picking up the missense mutations before, or what exactly is the reason, but we do have to look into that. And we have the other mutations also. We could pick up 7 novel mutations, and the youngest child which we could diagnose was 3 months old, and the oldest was 23 years old. This is in the pediatric neurology department. So there's definitely a need for registry, and we really need, coming from a developing nations, some cost-effective tools for identification of these disorders. So talking about the challenges, we have challenges from the clinician's perspective as well as the patient's perspective. Like I said, there is lack of integrated referrals and trained workforce. Resource constraints is what we talked about. We do not have facilities of MRI, MRS, also in certain cities also. And we are jack of all trades and master of none. So we are supposed to be seeing all pediatric neurology cases, and we have very limited time to dedicate to a specific specialty or a subject. But we do try to find time for that also. There has been undiagnosed cases due to cost of investigations, and there have been a frequent fallout. The moment they realize that they have to pay this much money for the testing and facility, and they know it's probably a genetic disorder, they usually fall out. For the patients, there is lack of awareness and low understanding. There is still no understanding of genetic disorders as such, and there is high consanguinity. Apart from that, there is always a difficult trajectory to reach the expert. There is longer wait until diagnosis. Affordability remains always an issue, and there is lack of patient advocacy groups, foundations, and genetic counselors. So talking about the average cost of testing and care in our country, this is like in a good infrastructure hospital, I would say. MRI would cost anything between $100 to $200. A genetic test would cost about maybe $300 to $350, and an array would cost around $90. And in totality, if I just tried to put a cost bracket to this, so it would come around $500 to $1,000, but they do not have that much also. And a regular clinician visit or clinician fees would be around $10 to $20, with a rehabilitation visit $10 to $20. So this much of affordability is also not there, and the government is trying to bridge this gap, but it's still way too far. So how can we contribute from the developing nations? We have a vast population, and there is a lot of case burden, and there has been intercommunity marriages and consanguinity adding to the case burden. So there is access to healthcare facilities in the larger cities. We are trying to reach out to our village population also. And there has been a gradual decline in the cost of the genetic testing, and more setups are gradually being built up. There has been reach out groups in the form of camps and peripheral clinics, and we have extended healthcare to our neighboring nations as well. One good thing is that we have a close-knit, at least pediatric neurology as well as neurologists working together for the registry and foundation. So I am sure that like Mario and Nicole have taught me, take small steps with each day, and we would reach somewhere definitely. Thank you very much. I would really like to thank all my mentors and my team at my country. Thank you.
Maybe 2 minutes for a round of questions. I think this was a great presentation. Thank you all. Questions? There's one all the way in the back.
Hi, I really enjoyed your talk, and I wanted to ask, as a patient advocate leader, how do you recommend we go about finding patients in our disease area? We have one patient in Bangladesh who reached out to us via Facebook, but is there a way we as patient advocates can be more proactive in trying to assist in your efforts?
Well, thank you very much for this because it's very nice to know that you are, you want to extend your support. Yes, I would say that if we have a, formal group or foundations, we still do not have any foundation. This is on my mind. I am very soon working on it to have, a leukodystrophy foundation, something similar to what the Western world is having, so that we can have one proper platform where we can actually reach out for help also, and we can collaborate on those grounds. And we are working on social platform also, like maybe, Facebook and all those groups where we can connect to our people as well as to the other parts of the world. Yes.
Allah.
Thank you very much for your talks. I also have a question for you. Is it your sense that if the barrier to genetic testing could be removed, if there were ways of offering, globally panels that were free or very inexpensive, would that remove the barriers to access, or would the barriers to getting to a doctor and then the implications of a genetic diagnosis culturally and then the post-care still be such barriers that it wouldn't make a difference? I guess that's my question.
Well, thank you very much, Adeline, for that question. But I do agree that there are multiple barriers, but genetic barrier is also one big barrier. And we have to start it at some point where we can go one by one as well. So I think if we can take at the genetic part, if we can take care of the genetic part, I think that would really probably take 50% of our problems away as well. So that is also a big, that would also be a big help, I would say. Yeah.
Yeah, one last question here. Over here.
So in India, you can't send DNA out to collaborate with research institutions, can you? So that is probably going to be a limitation.
There are regulatory bodies for that, but it's not that difficult. We have certain papers and all to be filled up, and certain government-related. We have a research body called ICMR, and we do have, we have to write to them. We have our ethical board meetings and everything. But once it's through the process, it could be done, definitely.
Because there are initiatives such as with Illumina and other companies. For example, we're involved in a project with India, with Bangalore, NIMS and AIMS to do that.
Yes.
It's a bit paperwork heavy, but if you're willing to do that, then that would be a distinct way of getting all of your patients analysed.
That would be
really nice. Of course, the interpretation is another matter in diverse populations. That's quite hard. Yeah.
We have a lot of genetic facilities also, but the thing is that uniformity is not there. Probably one genetic center may vary from the other in terms of analysis. So that is where, again, challenge comes. So, yeah, that would be nice, actually. Thank you.
Thank you.
Yeah.
All right, well, thank you very much. It was a great presentation. So we're going to take a